productions, okay, are currently being treated with these antibiotics compounds. Okay, projections of global consumptions of antibiotics in food, animal productions, assuming no policy changes, okay, is expecting to increase by 11.5% in 2030. Okay, for humans, the global use of antibiotics, okay, was estimated, okay, to be 40.2 billions, okay, defined daily doses, okay, in 2018, and the consumption rate has increased, okay, dramatically by 46% in the last 2 decades. Okay, this misuse and overuse of antibiotics have given rise, okay, to the serious problems of antibiotics resistance worldwide, and they are the main drivers in the developments of antibiotic resistance in pathogens. So what is the misuse of antibiotics? Okay, so misuse of antibiotics was defined, okay, as the misuse or overuse of antibiotics, okay, with potentially serious effects on health. And according to the Centers for Disease Control and Prevention, about 1/3 of the antibiotics used in human is not needed nor appropriate. And this overuse of antibiotics by healthcare sectors and livestock farming, as well as aquaculture industry, okay, not only exacerbated the antibiotic resistance, but also have led to environmental pollutions with antibiotics residues. Okay, and for your informations, okay, although most of the antibiotics, okay, were water soluble, okay, but they are not fully absorbed by humans and animals. And antibiotics are only partially, okay, digested and absorbed by the human body and animal. Approximately 30 to 90% of the antibiotics, okay, are not, okay, absorbed by human, whereas about 75% are not absorbed by the animals and eventually will excreted through urine and feces within 8 to 24 hours after consumption. Okay, this which can directly, contaminate and harm the surrounding environment, and the excretions rates of antibiotic is various. Okay, and some antibiotics, okay, has been reported that they can stay long, persistent in the environment, like antibiotics such as penicillin, okay, they have excretions rates of 60%, and others antibiotics such as fluoroquinolone, okay, they have lower values of about 30%. Okay, excretions rate. Okay. Okay, the sources of disseminations of antibiotics into the environment, okay, they have they have what we call several way. Okay, the sources could via which antibiotic can be released into the environment. Okay, this could be diverse and are related mainly to human activity. Okay, and the antibiotics excreted by human and animal, okay, commonly in their waste are largely, okay, are metabolites, and the conventional wastewaters and recycled waters treatments are only, okay, partially effective in uh, their removal or degradation. And the fates of antibiotics, of course, are dependent on their mobilities and persistency in the environment. Okay, the detected concentrations of antibiotic residues, okay, generally range from nanogram per liter to microgram per liter. And the residues of synthetics and semi-synthetics antibiotics, okay, they are more chemically stable and less susceptible to bacterial degradation, okay, processes. Antibiotics such as fluoroquinolone and sulfonamide. This is the reason why these antibiotics residues are often, okay, detected in the environment. Okay, and as a result, okay, antibiotic may reach different aquatic environment, and some environment, okay, such as the areas around the pharmaceuticals plants, animal farms, aquacultures, hospitals, okay, as well as those receiving anthropogenic input. Okay, these environments, they are constantly, okay, exposed to contaminants, okay, that containing the antibiotics. Okay, this resulting in the persistence of varying concentrations of antibiotics present in this environment. Okay, and these antibiotics, okay, they may become a source, okay, of nutrients for certain groups of microorganisms, and becomes degraded, means that they become the food sources, means the nutrients, means these antibiotics becomes a nutrient source, okay, for the bacteria. Only certain groups of bacteria are able, okay, to use up this. Okay, and of course the biodegradations, okay, depends on many factors. Okay, bioavailable fractions and the local environmental condition. Okay, of course this including compositions of the what we call the local microbial committee, the pH, temperatures, water content, organic carbon contents, and humidity. Okay, and non-biotics degradations sometimes can also take place. Of course, all these via hydrolysis, oxidation, reductions, or photolysis. And this also depend on many physiochemical properties and environmental condition. And One Health, okay, is a collaborative and transdisciplinary approach, okay, which intends to preserve human, animals, and environmental health, okay, through the surveillance, preventions, and mitigation. And the main objective of One Health is to attain optimum health and sustainabilities for humans, animals, um, and the environments at the same time. So antibiotic resistance is the best, okay, illustrates the One Health approach as antimicrobial resistance, okay, affecting humans, animals, and environment. And nowadays, antibiotic resistant genes and antibiotics resistant bacteria, okay, they have emerged as a new class of environmental pollutant, okay, in aquatics environment that are of significant concern worldwide, as they can promote the developments and selections of antibiotic resistance in the environment. Okay, so what is antimicrobial resistant? Okay, I mean that many of you may already know antibiotics are used, to treat or prevent some types of bacterial infections. Okay, antibiotics resistant occur, okay, when bacteria change, okay, in response to the use of the types of medicines. Okay, these bacteria, they're able to develop and have the ability to defend, okay, the medicines that designed, okay, to kill them. Okay, in which they are not killed and have the ability to continue their growth. Okay, we call it resistant. Okay, and resistant infections can be difficult, okay, and sometimes impossible to treat. Okay, in 2019, okay, AMR has been declared, okay, as one of the top 10 global public health threats, okay, facing humanities by WHO. Okay, and a global reviews, okay, on antibiotic resistance highlighted that if we do not act now, okay, to tackle AMR, infections that are resistant to antibiotics will kill 10 millions of people by 2050. Okay, Asia is one of the most affected region, and these figures is actually matching to the annual global death toll of cancers. Okay, and which equivalents to one person, okay, dying every 3 seconds and more dying annually than compared to cancers. Okay, and also researchers estimated that between 2016 and 2015, the world could expect to lose between 60 and hundreds, okay, trillions worth of economic output because of this AMR. Okay, this slide shows the developments of AMR and how they disseminate, okay, in the environment. Okay, antibiotic resistance is naturally occurring process. Okay, when microorganisms are exposed to antibiotics drugs. Okay, the starting point in these examples is a large bacterial populations, okay, where most of the bacteria are susceptible to antibiotics, but only a couple of the bacteria are antibiotic resistant by chance. Okay, when a bactericidal antibiotics is added, okay, which kill most of the susceptible bacteria in the population, while the resistant bacteria, of course, will survive. Okay, only the resistant bacteria will continue to proliferate in the presence of the antibiotics and increase their numbers, okay, over time. And the end results is a populations of mainly resistant bacteria. Okay, so it is important for you all to understand that selections of antibiotic resistant bacteria, okay, they can occur anywhere, okay, when an antibiotics is present, okay, at a selective concentration. And ARGs, okay, are horizontally transferred by mobile genetics elements, okay, between different bacteria cells. Okay, resistant bacteria, of course, they can survive, okay, under antibiotic treatment, and they can increase in numbers by natural selections. Okay, of course, mutation can also result in antibiotic resistance in bacteria in environment, and bacteria resistant to antibiotics, okay, therefore can be intrinsic or acquired. Okay, intrinsic resistance, okay, results from naturally occurring threats in a species. Okay, remember, yeah, so bacteria, okay, they can actually the resistance to antibiotics, okay, naturally means that intrinsic, okay. Acquire means the bacteria gain the antibiotic resistance after exposed to antibiotics, okay, or they obtain the resistant genes, okay, from others, okay, uh bacteria, or by viruses. And according to a new global studies, okay, our world's aquatic systems are widely contaminated, okay, with antibiotics. Okay, the antibiotics are extensively detected in surface waters from different aquatic systems such as sea, bay, rivers, lake, streams, and canal. A totals of, hundreds of 69 types of antibiotics has been detected, and their metabolites, okay, were also identified. Okay, so far, based on the literature review, 160 types, okay, of antibiotics actually has been detected in surface water, and whereas for sediments has reported 111 types, okay, of residues. Okay, and these antibiotics detected were can be categories into 6 classes. Okay, which including what we call sulfonamides, tetracyclines, okay, quinolones, beta-lactam, macrolides, and others. Okay, experts are expecting a 65% of increase in antibiotics concentrations in aquatic environments, okay, by 2050. Um, and wait. Okay, dangerous levels of contaminations, okay, were most frequently found in Asia and Africa. Okay, the safe levels can range from 20,000 to 32,000 nanogram per liter. Okay, depending on the antibiotic itself. Okay, the figure here showing the antibiotic residues that detected from the East and Southeast Asia country, And sulfonamides and tetracyclines are the most prevalent antibiotics that are found in aquatics environments. Okay, these are mainly due to their low cost and effectiveness against a broad range of infections. Okay, the predicted no effect concentrations is the concentration, okay, below which exposure to a substance is not expected to cause, okay, any adverse effects, okay. And antibiotics actually can be grouped by either their chemical structures or mechanisms of actions. Okay, they are often complex molecules that may possess different functionalities, okay, within the same molecule. Okay, because of their difference in functionality, their physical, chemicals, and biological properties, okay, their absorptions, behavior, photoreactivities, and antibiotics, activities, and toxicity, okay, all this may actually change with pH. Okay, so therefore, under different pH conditions, antibiotic can be neutrals, ionics and ionics or sweeter ionics. Okay. Mm, okay, what are the factors actually could affect the concentrations and profile variations of antibiotics, okay, in our environment? Okay, first is geography. Okay, because infectious diseases, okay, they can be varies by geographic regions and population, and they change over time. Therefore, antibiotics practices, okay, used by areas and country, okay, for the what we call the disease treatments and preventions, okay, could varies among the different country and regions. Therefore, like social economy factors, okay, like populations, uh, the death rates, the medium age, okay, the human health status, and the local unemployment, all these, okay, could greatly affect the type, okay, and the distributions of antibiotic residues, okay, into the aquatics environment. Okay, next is the phase distributions characteristics, like the distributions of antibiotics, okay, between the dissolved and particulates phases and between water and sediments, okay. These are important factors, okay, as well, that affect the contamination levels and the fate of these antibiotic residues, okay. And most of the studies, okay, they use membrane filtration, okay, as a sample pre-treatment step. Therefore, the measured concentration represented the dissolved phase concentration, okay. And the distributions of antibiotics between waters and sediment is also driven, okay, by the natures of the compound and the functional group of the antibiotics, as well as the sediment's properties. So therefore, when you refer to antibiotic residues concentration, okay, you may first check on where actually these antibiotics extracted from, okay, whether it's from waters or sediment, okay. Usually most of the time, okay, concentrations, okay, in sediments, okay, may greatly difference compared to water itself. Okay, I also highlighted that the in situ environmental parameter means the uh, physiochemical parameters, okay, is very important. And in situ environmental parameters like salinities, temperatures, moisture, okay, oxygen's availabilities, or what we call the soft oxygens, okay, of the environment itself also affect the partitioning's behaviors of the what we call the antibiotics in the environment. Okay, and for instance, like like the uh anthropogenic activities, okay, I mean nearby the aquatics environments, okay, also considers what we call a major source of antibiotics, okay, and different antibiotics, okay, the type of source of contaminants, okay, also difference among geographic region, okay, the antibiotics dosage, okay, used as well, and the how efficient of the wastewater treatments efficiency at each country, okay, could also be the factors that affecting the distributions and profiles in aquatics ecosystem. Okay, and of course, like other factors like climates factors. Can everyone mute your mics? Okay, like climatics factors like rainfall, okay, I means that temperatures, precipitation, okay, all this may also actually give impact to the antibiotic concentration by changing the water flow, okay, and affecting the usage and the biotransformations of antibiotics. Of course, during the rainy seasons, okay, we will experience a high flow, okay, and this will dilute the antibiotic concentration, okay, in the water itself. Of course, during high flow, I mean high rainfall or seasons, it also wash away uh, surface, okay, this will cause a high surface runoff. All this will actually contribute to the concentrations of antibiotic in our aquatic system. Of course, low flow usually happening during the dry season, and biotics factors, okay, also is important in affecting the profiles, okay, of antibiotics in the environment, like bacteria's community structures, okay, the microbial abundance, okay, their biodiversity, and the microbial process rates, okay, will also play a role in the degradation, okay, degradations of the antibiotics, okay, in the environment. And the antibiotics, okay, of course, as a pollutant, okay, they will pose potential ecological risks, okay, to aquatics organisms and human health, okay. And most of the available studies on antibiotic contaminations or surface waters, okay, included ecological risk assessment, okay, using the risk quotients, okay, using the RQ scheme on non-target aquatics organisms like algae, invertebrates, okay, some aquatics organisms, okay, like fish, okay, um, in measures of the impacts of these antibiotic residues into, on our ecosystems. Okay, And the risk questions of an antibiotics in waters, okay, to certain species, targeted species, okay, are estimated by dividing the measured environmental concentrations by predicted non-effect concentration. Okay, and these so-called PNEC values, okay, are actually based on toxicological data, okay, okay, and have maximum lethal concentration. And the levels of risk are generally classified into 4 groups. Okay, no risk, low risk, medium risk, and high risk. Okay, no risk is means the acute value is less than 0.01. Low risk is less or equals to, I mean, between 0.01, okay, to 0.1. Okay, medium risk is between 0.1 and 1, and high risk is more than 1. Okay, and study has revealed that the antibiotics in many aquatic systems, okay, throughout the world, okay, inhibited ecological risk at different levels, okay, to aquatics organisms. Okay, and some of the antibiotics, for instance, okay, researchers have found that like sulfonamides, macrolides, okay, and beta-lactam, okay, they exhibited higher ecotoxicities to aquatics organisms compared to tetracyclines and quinolone. Okay, at regional scale, okay, the highest risk, okay, potentials of what we call antibiotics, okay, were found in Africa waters, okay, followed by Asian. And researchers also found that cyanobacteria and algae, okay, they are the most, okay, sensitive to antibiotics among the organisms of different trophic level. And if let's say the antibiotic pollution is getting worse, okay, the elevated antibiotics residues, okay, may negatively alter the balance, okay, of the aquatic ecosystems through the I mean deleterious, okay, effects on natural microbial communities, okay, and on algae, the primary producers of aquatic food webs. Okay, antibiotic residues, okay, they are not only causing the pollution in the environment, and these pollutants are related to the developments of antibiotic resistant gene and antibiotic resistant bacteria, okay, which has been considered as the most critical risk, okay, regarding this what we call micro pollutant. Okay, and aquatic environments are important reservoir, okay, as they may represent the origins, okay, amplifiers, and or what we call the reservoirs of the environmentals, humans, and or what we call animals, antibiotic resistant bacteria and their genes, okay, as well as bioreactors, okay, that facilitating the emerging and disseminations of ARG transfer, okay, between the pathogenic and non-pathogenic bacteria. And remembers, okay, when the release of antibiotics, okay, compound into the environment, okay, this actually allows, okay, these antibiotics to actually have direct contact, okay, with the naturally occurring microbes and act as a driving force, okay, for microbial evolutions and emerging of more resistant bacterial strain. Okay, and the selective pressures or cost exerted by these antibiotics could actually determine the occurrence, amplifications, and the disseminations of the resistant gene, okay, in the environment and pathogens as well. Even low concentrations of antibiotic actually can result in the selections of what we call the ARG, okay, making the what we call the establishments of a safe concentrations of any antibiotics compound in the environment at challenging tasks. Okay. And bacteria are able to respond to selective pressure, okay, and adapt to new environment, okay, by acquires new genetic trait as a result of mutation. And horizontal gene transfer is the most involved mechanisms, okay, for the disseminations of resistance in aquatics environment. Therefore, you need to know what are the key mechanisms, okay, involved. There are four mechanisms of horizontal gene transfer in bacteria: transformation, transductions, conjugation, gene transfer agent, okay? And besides bacteria, also, they develop, okay, their own defense strategies, okay, against these antibiotics, okay, that call resistance mechanisms. And the main mechanisms of resistance are important for these bacteria, okay, to help them to actually limit the uptake of the antibiotics. Okay, these bacteria could also active their efflux, okay, pumps, okay, of an antibiotics. They also have what we call enzymatic alterations of antibiotics or modifications of an antibiotic targets. Okay, antibiotics sequestrating by protein binding, overproductions of targets, okay, metabolics by bypass, okay, of targeted pathway, okay, all this can actually inactivate of an antibiotics, okay, and these mechanisms, okay, may be native, okay, to the microorganisms, or they actually could acquire this so-called mechanisms, okay, from others microorganisms as well. Okay, nowadays, okay, using the molecular techniques, okay, many studies actually have shown that the prevalence of different ARGs, okay, and ARBs in our aquatic ecosystems. Okay, when we think about ARB according to the One Health concepts, okay, the aquatics environment is important, okay, as the human animals environments interface, okay, and the water's environment means our aquatic systems, okay, they actually plays an important role as a sink, okay, reservoir for ARB and for antibiotic resistant gene, okay, from the human activity or what we call the anthropogenic activities, and they are important for linking the bacteria and gene, okay. So, and the first multi-drug resistant plasmid PRA1 actually was isolated from the fish pathogens, okay, and the plasmid structures backbone, okay, the backbone, okay, has been found and detected in others marine fish pathogens and human pathogens, uh, conveying different AIGs cassettes. Okay, and these findings actually give evidence, okay, a strong evidence, okay, suggests that the aquatics environment actually are suitable for the persistent transfers and dismissions of ARG. And I mean that it is well known that majorities of the aquatic environment bacteria are unculturables or yet to be cultured. And I mean that, as I mentioned in the class before, okay, the conventional culture dependent method, okay, for monitoring ARB and ARG can only reveal, okay, very low percentage. Researchers estimated only 0.1 or less of the true aquatics bacterial communities has been revealed. Okay, and through the quantitative PCR method, okay, I mean the antibiotic resistant gene were found in high relative abundance, okay, in non-culturable bacteria than culturable bacteria. Therefore, unculturable bacteria actually are the major reservoirs of antibiotic resistant gene in aquatics environment. Okay, and to better understand the anthropogenic impacts on antibiotic resistance, okay, our laboratories have selected a river in Malaysia, okay, the Larut River in Perak, okay, as a study models to evaluate the impacts of discharge from hospital, zoo, slaughterhouse, okay, to emergence of sulfonamide resistant bacteria from the aquatics environment. As sulfonamides are the oldest and remain among the most widely used antibiotics. Okay, from our studies, okay, our research shows that the microbial communities actually was significantly shaped by the sulfonamide residues. Okay, and the key antibiotics detected in these studies, okay, especially in the hospitals, okay, exhibited significant correlations with the microbial communities and the total surgeon, okay, and in the slaughters effluents, okay, we found that adenobacterius and gamma proteobacterius, okay, shows a positive associations, okay, with two types, okay, of sulfonamides antibiotics, and the differences in response by the observed microbial communities to sulfonamides, okay, antibiotics. Okay, these were probably due to the developments of antibiotic resistance mechanisms, okay, in the indigenous bacteria under the antibiotic selective pressure. Okay, and the CCA results, okay, in these studies, okay, also shows that the microbial communities actually significantly shaped, okay, by the physiochemical parameters, for instance, salinity, DO, okay, COD, phosphate, and total nitrogens, okay, and these findings actually is, consistent with other previous studies that the changes in the abundance and compositions of the microbial bacterial communities, okay, can be attributed to in situ physiochemical parameters. Okay, in these findings, okay, we found that salinities and DO, they are the important drivers, okay, of the bacterial diversity and composition. Okay, and these studies conducted in Malaysia's rivers, okay, clearly shows that the sulfonamides residues and inorganic nutrient actually exerted significant impacts on microbial communities and on the resistant gene as well. Okay, and in this in these studies also I mean, through the network analysis, we have successfully identified 11, okay, potential surging bacterial carriers, okay, that can serve as a reservoirs of surgeons and play a role in disseminates those resistant genes among the microbial community in this river system. Okay, and then I mean nowadays through the reasons you know developments of the technologies, especially the NGS, okay, DNA sequencings and the metagenomic approach, they actually have further advanced our knowledge in understanding the role of microbial communities, okay, and the behaviors of ARG in the environment. Okay. So considering the central role of the water body in the development and spreads of AMR, so the active monitorings of antibiotic residues in the environment and control of disposals into the environments are really needed, okay, to actually help to control or to reduce, okay, the rates of AMR emerging and developments in our aquatics environment, okay. Therefore, I mean, it is important to have the environmental monitoring, okay, and surveillance and further research, okay, uh, to provide more data and evidence, okay, and better targets intervention. Of course, we also need to increase global effort, okay, to improve the integrated water management and promote water sanitation and hygiene, okay, to limit the development and spread of AMR in our environment, okay, of course, at the same time to reduce the infections and need for antimicrobials, okay, and it is also important to create robust and coherent national level governance, okay, planning, regulatories, and legal frameworks, okay, as well as establish coordinations and collaborations mechanisms. Okay, and we also need to have what we call international standard, okay, for what constitutes good microbiological indicators of AMR from environmental sample, because different country or region, they have their own standard, okay, for uh safe antibiotic concentration, okay. Of course all these, okay, standards is important to use, okay, to guide risk reductions decisions and help also to create the effective incentive, okay, to have such a guideline, okay, to actually reduce, okay, AMR, okay. I think that's all for today's. Okay, that's all, and I will, okay, see you all again, okay, at 2 o'clock. Okay, any questions? Okay, if no question, thanks. Yeah. Okay, bye. See you later.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.